Hello everybody and welcome to today's webcast. Why is the sterility of your drug product much more than just a test of sterility? I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Nelson Labs and Sterigenics. Sotera Health business entities, Nelson Labs and Nelson Labs Europe offer microbiological and analytical testing and consulting for medical devices, implants, and pharma products. Sterigenics provides contract sterilization solutions for medical devices, pharmaceutical, and high-performance materials. Our speakers today are Anik Gillette and Martel Winters. Anik is Technical Director, EO Pharma at Sterigenics. In her role, she leads the ethylene oxide sterilization projects at numerous Sterigenics facilities and supports the facilities as the technical expert with pharma project responsibility. A seasoned speaker, Anik presents widely at industry conferences, often on innovative and customized solutions for terminal sterilization of pharmaceuticals. Martel is the Director of Scientific Competency at Nelson Labs. He's a specialist microbiologist and a radiation specialist. He's been at Nelson Labs for 25 years and spent his earlier years working in the radiation sterilization group responsible for bioburden testing and radiation validation studies. His specialties include microbiological process validation and radiation sterilization of med devices, allograft tissue, and pharma products. And today, Anik and Martel are going to discuss sterilization and why it is a pivotal step in the pharma drug development process to help ensure overall safety is maintained. We will be accepting questions throughout the talk. You can send those in using the box at the lower left of your screen, and questions will be answered after the presentation in our brief Q&A. And this presentation will be archived on our website, contractpharma.com, for 1 year. If you're experiencing any tech difficulties, you can type those into that box as well, and someone on the back end here will help you out. At this point, I'll turn it over to you, Martel.
Thank you. It's nice to be able to have this interesting discussion on this topic. Anik and I discuss these topics quite a bit and find them quite interesting, so hopefully you will as well. So when we talk about sterility and drug products, a really big part of achieving sterilization is very much like a game of chess. There's a lot of strategy involved. There are many opportunities to have the end, to use the end goal to help drive the activities that you do in the earlier stages of the drug development process. And so I look forward to sharing some of these concepts regarding sterilization and sterility assurance and the strategies that we try to employ to get the most success as quickly as possible. When we talk about the term sterility assurance, first on the left you see what it is not. So sterility assurance is not a sterilization cycle or a BI test result or a product sterility test result. Although these are components of sterility assurance, they're a small component. And really, sterility assurance is an aspect that is addressed in all stages of a product life cycle. So the product design, R&D, manufacturing, packaging, and then sterilization. So sterility assurance is meant to be an all-encompassing aspect of the product life cycle. For some examples, I guess first, when we talk about sterility assurance or assurance of sterility, there's a relatively new document from ISO. It's Technical Specification 19930. And when we talk about assurance of sterility or sterility assurance, we're talking about really a qualitative concept comprising all activities that provide confidence that the product is sterile. So I have underlined all activities that provide confidence the product is sterile, which means that this goes back to reinforce that sterility assurance or assurance of sterility is not just a sterilization process, it's all activities that provide that confidence. So when it comes to this thought process of sterility assurance, when does it start? So those of us that are sterilization people, Anik and I largely are dealing with products as they show up on our desk. Company, we receive products and they say, we want this to be sterile. And usually that's when we get involved in the sterilization process, or in sterility assurance. But really we prefer that sterility assurance start much earlier in the product design stages. So if it's a reusable product, for example, if we're talking devices, then the question is, are they designing products so it can be cleaned? For single-use products, whether they be devices or drugs, the idea is, can the sterilant reach those critical surfaces? Is the product designed to allow for sterilization? And then very quickly after that comes the R&D side, where you're looking at, as you're selecting materials, chemicals, ingredients, can you select ones that are sterilization stable? And these two steps, product design and R&D, we find that these are the two steps where sterilization tends to be considered least and where it really should be considered most. When it comes to manufacturing, sterility assurance on the manufacturing side, what kind of microbiological controls are in place, both as far as the actual manufacturing, but related to manufacturing at your suppliers. It's easy to demonstrate or to structure really good microbiological controls over what you are doing at your locations, but it's frequently underestimated the criticality of understanding what microbiological controls your suppliers have, because those components or those ingredients that you're purchasing from suppliers can play a significant role in the final microbiological status of your product. Packaging, I think packaging is one place where sterility assurance is often considered quite frequently. I think generally speaking, in the packaging phase, many manufacturers are considering the sterilization and ensuring that the packaging is enabling sterilization rather than inhibiting. But I still occasionally do see situations where we have a product that is intended to be sterilized, for example, with a gaseous sterilization method, and there's very little breathable space on the pouch, which does make that more complex. And in sterilization, the last one there, that's, I think, generally people tend to equate sterilization with sterility assurance, and so I don't add much more there because that's already a pretty important mindset. What we're trying to accomplish here is getting that thought process of sterility assurance considered much earlier in the process. So, sterility assurance concepts. Generally, when we talk about the word sterile, if it is, I think oftentimes the sterile is equated with sterility assurance level, which is the typical sterility assurance level is 10 to the minus 6. And although there are other designations of sterile, we'll get into that later. So the SAL is the likelihood of surviving microorganisms. You see all these different SALs there, 10 to the minus 1 through 6, and 10 to the minus 6 equates roughly to, conceptually, to 1 in a million of having a non-sterile product. The actual definition of SAL is the probability of a single viable microorganism occurring on an item after sterilization. So, in the ISO 1099330 document, I have both what was in the original, what was in the draft, and what ended up in the final on the left-hand side, just to help clarify when we use terms lesser or greater, or what we mean by that, naturally 10 to the minus 6, oftentimes people will think that 10 to the, for example, that 10 to the minus 7 is greater than 10 to the minus 6, but it really is a lesser probability. If you think of what 10 to the minus 6 means, it's point, a whole bunch of zeros, and a 1. So as I go to 10 to the minus 7, I'm adding one more zero. The number is actually a lesser number. So oftentimes when we want to make sure we're using the terminology correctly, that 10 to the minus 7 is a lesser probability of a surviving microorganism and a lesser number, but it does provide, as it says over on the left, it does provide increased assurance of sterility. So it can be tricky sometimes when we're using terminology to make sure that we're being clear in our terminology, our description of those differences in numbers. So why 10 to the minus 6? 10 to the minus 6 is, when we're talking about terminally sterilized products, that tends to be kind of the gold standard. And like I mentioned, it's roughly it means a 1 in a million probability of non-sterility. But where did that come from? 10 to the minus 6 is this magic number that pretty much the whole world refers to. But where did that come from? Why is it selected? What is the purpose behind that? So there's likely, I was not involved in the sterility assurance industry back in the 50s and 60s. I think clearly there was some guidance or drew some concepts from food processing. In 1970, the Nordic Pharmacopoeia selected 10 to the minus 6, and you see the quote there, the translated quote, that the conditions that aim at such a result that in 1 million units there will be no more than one living microorganism. And so this is because they knew, as in the food processing, they understood that you don't step from non-sterile to all sterile. It's all a function of probabilities. And we'll go into that a little bit on the next slide. So in the Nordic Pharmacopoeia, they selected 10 to the minus 6. And pretty much everyone else in the world said, that sounds great. Let's do it. 1 in a million sounds like a great number. And historically, it turned out to be a good target value. In the 1960s, NASA and other worldwide space agencies selected different values. For example, 10 to the minus 4 is for spacecraft under certain circumstances. That was their target value. So 1 in 10,000 probability. But there really is no scientific rationale behind why 10 to the minus 6 is the correct number. That was not based on science. It was based on it's an arbitrary value that people felt many years ago provided safety. And it wasn't until that there were two very good articles published in 2012 and 2013 from Srun and several others and they found that it's not really the device or the product would need to be sterilized only to about 10 to the minus 3 in many situations before it would start to impact patient safety. So really, what's really necessary is somewhere between 10 to the minus 3 and maybe 10 to the minus 4, and the other is just additional safety. There are so many other factors that play a role in patient safety that an SAL value generally does not play a role in patient safety until you hit those higher numbers of 10 to the minus 3 or 10 to the minus 4. So just to show a little bit here, for those that are more visually oriented, we have a table here on the left-hand side. You have bioburden or sterility assurance level. So at the top, you see 1 times 10 to the 6, so 1 million CFU, and then scaling down to 10 to the 0, which is 1 CFU, and then you see it does not go from 10 to the 0 to sterile. It goes from 10 to the 0, then to 10 to the -1, -2, etc. So then instead of saying we have an average bioburden of 10 CFU per device or per product, then you get below, you get 10 to the minus values, and you're saying out of 10 samples, 1 of them will be non-sterile. Or out of 10 samples, or out of 100 samples, 1 of them will likely be non-sterile. So we get into these probabilities. So where do we actually perform testing in this scheme here? Generally, we are performing, when it comes to determining proper sterilization cycles, we're always performing testing somewhere in that range of 10 to the 0 to 10 to the -1, maybe close to 10 to the -2. We're testing at those values, and we do not test product at 10 to the -6. So how do we know that works then? The idea here, the reason I have this death curve essentially, so you see that this curve represents microorganisms. If we start with a high value of 10 to the 6, as you apply more sterilization, you see in the bottom it refers to either dose, if it's radiation, or time, if it's another sterilization method. As you apply dose or time, the microorganism count decreases. As I add more sterilization, I get more decrease. And we actually never want to perform testing at 10 to the -6. In order for there to be any statistical or scientific relevance at testing fully sterilized product, we'd have to test at least 1 million samples, and or more, depending on how you run your statistics, which is not feasible, clearly. So we do not test at 10 to the -6. We test that partway down this death curve of microorganisms, and then we extrapolate to the final sterilization, the final sterilization process. So on the next slide here, we have a case study that Anik is going to go through. Anik?
Yeah, thank you. Thank you, Martel, and yeah, great preliminary explanation. So yeah, we just wanted to illustrate what Martel just explained by giving you some practical examples. So here you see a fractional cycle, so it means that's a cycle with a short exposure dwell time. So here we selected 30 minutes. And these are the results we obtained for that cycle. So you see that we have no growth for the product itself, so for the natural bioburden. We have external challenges with a higher resistance, so with 12 positives, and the sport product or the internal challenges are in the middle with 4 positives on 20 tested samples. So on the curve below, so that's the same curve as Martel presented earlier, you see that we are well in that area. So we have some partial growth in our challenges. So that's what we want to obtain, because it allows us to calculate the D value. So it means the time we need to reduce the PI, the contamination, the population of microorganisms, of 1 log. And here, just for the example, we calculated at 6 minutes for the internal challenge, so for the product, and 7.5 minutes for the external challenge. So it means that if we extrapolate to reach the 12 logarithmic reduction, which would give us the sterility assurance of 10 to the -6, we would need to run a cycle of 1 hour and a half minutes, and that will give us the proper sterility assurance level. So that's how we calculate, how we extrapolate the cycle time to reach the sterility assurance level. So back to you, Martel.
Great, thank you, Anik. So this really helps to illustrate the importance of extrapolation. We're testing at these shorter cycles and then using math with a good history of experience to extrapolate to the final sterilization process. So this question of, do we ever test fully sterilized product? When might it be appropriate to test a fully sterilized product? So there is a document from AAMI and ISO. This is the Sterility Test document 11737-2, 2019. In this newer version of this document, we were careful to add details regarding this question. So in the introduction, we have a sentence in there. It says, excuse me, performing a test of sterility on product that has been exposed to the complete sterilization process provides no scientifically usable data and is not recommended. And in the annex, we have a similar comment there, that tests for sterility are not appropriate for confirmation of sterilization process effectiveness. So when we say tests for sterility, we're talking about at the full sterilization cycle. So not appropriate for confirmation of process effectiveness, sterility assurance level, or attributes associated with sterility, such as package integrity or shelf life. There's a really good resource here, an article published by Elaine Daniel and a handful of other people in the industry, called Product Sterility Testing: To Test or Not to Test? That is a question. And that helps to describe more detail behind this situation. So we really do try to avoid testing fully sterilized product, because there are no scientific data you can gather from testing a handful of products that have been sterilized to achieve 10⁻⁶. So there's another document in the U.S. It's AAMI ST67. This is a 2019 version, just came out last year. And this is regarding selection of an SAL, of a sterility assurance level. It has been a U.S. national standard since the first version, since 2003, and recognized by the FDA. So it proposed a formalized approach to what we've always done in the U.S., which is 10⁻⁶ or 10⁻�3, that either can be appropriate based on the patient contact. It talks about terminal sterilization being preferred to aseptic processing, and we'll get into more detail on that. And that this concept of selecting an appropriate SAL really is a risk assessment, rather than always the default being 10⁻⁶. Now, in most cases, that is still a target, but there are situations that are amenable to other values. So in the revision, we have additional information now regarding guidance on selection of an alternate SAL, something other than 10⁻⁶. More discussion on aseptic processing and how it correlates or does not correlate to SAL. There's some discussion on SAL versus PNSU, so probability of a non-sterile unit, and also modification, so product modification strategies to be able to achieve an SAL 10⁻⁶. Whether that be modification to product or also modification to the sterilization cycles to be able to achieve 10⁻⁶. That's still the target value around the world, and so the goal would be to achieve that if it's a type of product that should have that type of level. Then in 2017, there was an ISO standard on a very similar topic. So 19930, there's the title there. It's very long, but the idea is if you can't hit 10⁻⁶, here's a risk-based approach to trying to figure that out. So it provides, again, strategies, alterations you can do to achieve 10⁻⁶, but considerations if 10⁻⁶ cannot be achieved. So this is an ISO document meant to be worldwide. ST67 is more focused on it's a U.S. standard, but this is, of course, an international standard. And one comment that I want to draw out from that ISO standard is that it is not appropriate to compare an SAL for terminal sterilization with a rate of occurrence of a contaminated unit estimated by process simulation for an aseptic process. So you really cannot compare an SAL to a contamination rate or anything else you derive during the course of an SAL. I know you cannot read anything, you probably cannot read the details of that flow chart, and it is not the intent that you be able to read that, but I just want to make you aware there is a flow chart that's in the document that I think is very useful to go through this thought process of terminal sterilization versus aseptic processing or aseptic assembly. Okay, so I will turn the time now over to Anik.
Yes, so let's dig into more details about the two strategies that are possible to obtain or to produce a sterile drug product. And so basically you have two possibilities. So one is aseptic assembly, and the other one is terminal sterilization with different possible technologies like heat, radiation, gas, etc. So we'll go through them a bit more into the details after. And also we'll compare the two strategies. So here about aseptic assembly, you see a kind of summary on how this method is working. So you see the different components of a drug product. So the container, the drug itself, the excipients, etc. All these components are sterile already by different techniques, and then they are assembled together to produce the sterile finished product. So aseptic assembly is really a question of maintaining sterility of something. So that's really maintain something that has been maintaining the sterility that has been previously sterilized. For the terminal sterilization, you immediately see that it's a totally different approach, because there we have the different components that are not sterile, and they are exposed to a special process, and then you obtain the sterile product. So here that's really an effective exposure to a physical or chemical agent for predetermined extent of treatment. So on one side we have maintaining something, and on the other side we have an active exposure to an agent. So we will go a bit deeper in the comparison of these two approaches by explaining a bit more about them. But firstly, we would like to have your thoughts on that differences, and ask you the following question that I will let Martel to explain more.
Okay, so here's the first. We have another polling question a little bit later. So let's go through this question. The question here is: which statement is true? A sterility assurance level less than or equal to 10⁻⁶ can be obtained by, and you have a few options there. So let's take a moment, and if you will answer that question, and we'll give you just a minute to answer that, and then we'll discuss it. Okay, I think we have a decent number here. It looks like the quantity of people that are providing their answer, I think it's slowed down a little bit. Oh, so we got some more coming in, so we'll give it just another moment. Okay, let's go and stop here. So I'm going to make these results available to you, to the audience here. Okay, so what we're seeing we see about, so how can you achieve a 10⁻⁶ SAL? Is it through terminal sterilization and aseptic assembly? We still have more coming in here, but we're still seeing about the same situation where about right now about 50% of the people feel that you can obtain an SAL 10⁻⁶ by both terminal sterilization and aseptic assembly, and about 42% feel that it can be only by terminal sterilization, and then about 8% by aseptic assembly. And so this is a really important thing to understand, that when an aseptic process validation is performed, so we're talking whether you call it a media fill or a process simulation, when you are performing that type of validation, that those results are not used to calculate a value, to calculate a number. Those results are only used to validate that the process can be carried out without the addition of microorganisms. So the correct answer here really is number 2, that an SAL, a sterility assurance level, can only be obtained by terminal sterilization. The aseptic assembly does give you assurance that there is sterility afterwards, but as Anik just described, it is not applying an active elimination or inactivation of microorganisms. So there is no value that you apply after aseptic assembly. Okay, Anik, I'll turn it back to you.
Yeah, good. Very interesting survey. I think that's really interesting to see the different results. And definitely, I hope the rest of the presentation will help you to understand why really the answer 2 is the right answer. So here, really, that's to illustrate that and to ask you the question: do you really consider aseptic assembly and terminal sterilization as equivalent technologies? So, of course, both are able to give sterile product at the end. But the way to go there is totally different. So we will explain more. But anyway, it's true that the terminal sterilization is the preferred method by far. So in any standards, here I took the example an ISO standard, but it's the same in any other standards that you could find in the US or in Europe, that you have to use terminal sterilization when it's possible. So let's try to understand why it's the preferred method. And here, Martel explained already the concept of terminal sterilization and to obtain that SAL of 10⁻⁶, and that's really based on a validated method. And practically in the terminal sterilization world, we go far below. So it's not infrequent to go 10⁻⁸ or even 10⁻20. So it means that we have a lot of safety margin compared to the 10⁻⁶ we have to achieve. So let's understand the concept of the validation for that terminal sterilization. And that's really the basic concept that will help you to understand how it can be used for the validation. So if you see that picture here, that's really a pyramid with the hierarchy of resistance we try to obtain during the validation. So you see at the bottom the product bioburden, and above that you have the internal challenges and the external challenges. And the idea during the validation is to demonstrate that your external challenges are more resistant compared to your internal products, your internal seeded products usually, so the IPCDs, and more resistant compared to your product bioburden. So at the end, when you can show that your external PCDs have been sterilized, then you can assess that your product bioburden has been killed also. And how do we show that? We use sterility test. So that's the moment we will use sterility test during terminal sterilization validation. So that's to demonstrate that the product bioburden is deactivated, is less resistant compared to your PCD challenges. So that's the concept for the terminal sterilization. For the aseptic assembly, there really you cannot calculate any SAL. So you cannot calculate any sterility assurance level because it relies on multiple factors. So here I listed some of them. You see, for example, the equipment, the environment, the initial contamination of your raw material, the aseptic techniques. I could add also the technicians, etc. So really, you cannot calculate anything. What you can do is to estimate or to simulate your process to estimate your contamination level. And it's done through media field test. So that's where we will use some sterility test during aseptic assembly. So that's to test some samples that have been produced using the aseptic assembly process. So here again a question for you, and I will ask again my dear colleague Martel to ask it to you and to explain it. Martel, if you would be so kind.
Excellent. Okay, so another polling question here for you to provide answers. Which statement is true? During a batch review, a product test of sterility where no growth was detected. So this is a sterility test was performed and no growth was detected in that sterility test. What does that tell us? So of those three options, what do you think that tells us? We'll give you a moment to review that and provide an answer. Okay, I think we might have most of the answers in, looks like. So we have, we'll send these results out here. We're getting a few more in. So I'll send these to the audience and then we'll talk about it as people continue to answer. The vast majority are selecting number 3, which is information amongst other information that evaluate the sterility of this batch. So even in the case of aseptic assembly, this is really the case. So in aseptic assembly, you have all these other aspects of the process that you are monitoring or controls you have in place, and those controls have to be verified to be in place. The monitoring results have to show up being okay. These are often environmental monitors and monitors of raw materials and perhaps parts. It's product that's selected throughout the process occasionally. There's a lot of other information that goes into determining that the batch is sterile for an aseptic process. So the product sterility test really is just one small component of all this other information that the batch is sterile. All right, Anika, I'll hand it back to you.
Yeah, thank you for these explanations, and yeah, it seems we already made a good job explaining this. So the correct answer indeed is the third one. But let's continue the presentation and really explain the purpose of the sterility test and what they really mean in the different way they are used. So here you see the 3 uses of the sterility test. So the first one is used, as we said, during process simulation, so for the aseptic assembly approach. The second use of sterility test is during the validation of a terminal sterilization process. And the third occasion where you will do sterility test is for QC release. So the 2 first uses are really to assess a process, and the third one is really to assess the product. So these are the 3 cases. So you see the test itself is performed exactly the same way. So only the way we will use the data are different depending on the purpose. So hope it clarifies a bit your minds, because there are a lot of confusions around that. So we also wanted to propose you a practical example of a test of sterility that is performed in aseptic assembly. So here in that example, very, very simple, but that's a batch of 100 milliliter. And the idea is to produce 100 vials of 1 milliliter. So according to the USP 71, we need to test 10 vials. And in this example, we consider that the stock solution has been contaminated with 10 microbes, so with 10 CFUs. So let's think a bit deeper into that and understand really how the test is performed and what it does really mean. So you took your 10 samples, but you cannot have any evidence that your contamination is equally distributed. So you do not know if the 10 samples you will take could be contaminated or no. And even you don't know if one bottle, if contaminated, will contain 1 or more than 1 CFU amongst the 10 that we had in the beginning. Even if you have that sample, you have that contaminated bottle, contaminated vial, you are not sure that it will be able to be viable, so that you can detect it, that you can see it, that it will grow in your media. So definitely the probability of having a false negative is high, and you cannot calculate that. So that's why you cannot calculate the SAL as we would do for terminal sterilization. So it's very important not to, I'm missing a sentence here, but it's very important not to overestimate the use of or the results of your sterility test. And if you look at the ISO standards, it's even not recognized to release your product. So it's not okay to use a sterility test to release your batch. So you cannot do that because of what I explained before. Because it does not give you so much value. However, the EU GMP still does require that. So, and that's why it creates a lot of confusion amongst the pharmaceutical industry, because in most of time they are obliged to do these tests, even if the method is fully validated to a sterility assurance of 10 to the -6. In the US, I know that there are some possibilities not to do that, and depending on the situation, you can do some risk analysis and avoid to do that. But hope you understand now that it's not needed to demonstrate that you have a sterile product with the required level of sterility assurance. The next part of the presentation is about the selection of the sterilization methods. So we say that there are plenty of them, different of them, but definitely you should select first the terminal sterilization methods. The first one to consider is the dry or moist heat, because it's the most convenient and very cost efficient. But a lot of pharmaceutical products cannot resist to high temperatures. So that's why the second option would be radiation, and then other terminal sterilization techniques like gas sterilization, EO sterilization, so ethylene oxide sterilization, for example. And only if these methods are not suitable for your product, only after having considered them, you can go or think about aseptic processing and potentially with additional filtration step. But it's very important to keep in mind that you should firstly think about terminal sterilization method before going to aseptic assembly. And based on a survey published by the PDA 3 years ago now, but I think the situation is not so different nowadays. We had, amongst 75% of the pharmaceutical industry manufacturers were using aseptic assembly, and amongst these 75%, 30% could use terminal sterilization for their product, where they were now produced by aseptic processing. So you see that we still have a lot of margin or efforts to do to explain that concept, and also to make people understand why this is not the same of doing a sterile product using terminal sterilization or using aseptic assembly. To do that, you can do a structured approach with a kind of screening approach. So that's something we can definitely help you with at Sterigenics and with Nelson Lab. A bit of words about the different possibilities for terminal sterilization. Ethylene oxide, irradiation. These are the two most common industrial methods. And then you have, as I said, most heat, dry heat. You have also VHP, gas plasma. And we see also now new methods like nitrogen dioxide, which is a technology we can propose you now also at Sterigenics. So prior to making your choice, it's not an excuse to say that your product is not adapted to go for terminal sterilization. So it's clear also in the standards that you should consider the different components of your product to see if they could resist to terminal sterilization. So maybe you can adapt your formula, maybe you should change your container, or maybe you should select compatible components. So these are things that could be done when you consider sterilization at the very early stage of product development. So as said by Martel at the very beginning of the presentation. So this is a first thing to consider. And then also, once you go for the terminal sterilization, you should know that there are a lot of mitigation possibilities to be able to use your sterilization process and to optimize it to make it suitable for your product. So I give you some examples that will be quite quick, but of course, if you need more details, do not hesitate to contact us after this webinar. So here for radiation, for example, most of people think that only the 25 kilogray, so the VD max method is the only possible method. But you can use lower radiation dose. You can also use deep freeze process, or you can also inert your packaging. So these are things that could allow your product to be sterilized by radiation. The same for ethylene oxide. You could optimize your cycle design. So again, it's very popular to use the half cycle approach, which is making very good results with good safety margin. But it means also that frequently you have long cycle at high temperature with high EO concentration. But we have possibilities to really optimize that, and even to run this process at low temperature, so at body temperature, 38 degrees Celsius. Maybe you could also choose some alternate methods. So I said about Noxylizer, that's a new technology. There you can, it's really promising, or it's giving very good results for the pre-filled syringes, for example. And it's good because it's a technology working at ambient temperature. With the residuals are not as toxic as as we could have for ETO. It's just not appropriate for cellulose. So then you probably have to make some adaptations to your packaging. Also, for terminal sterilization, as Martel explained also earlier, you could maybe select higher SAL. So, for example, 10 to the -4 or even 10 to the -3. So again, that's based on risk analysis, and of course, for that you need to have the approval of the authorities, but that's feasible. So you cannot say immediately, my product cannot support terminal sterilization. There are many options that could be possible for your product. And here are some examples of products that are currently sterilized in our facilities, pharmaceutical products that are sterilized. So first example, that's nano suspensions. We also have a lot of lyophilized products, dry powder and APIs, and we have more and more of these. For ethylene oxide, we have also a lot of APIs, pre-filled syringes. I said about that, but that's for mainly external sterilization. We also have active implants, auto-injectors. So all these are combined products. So we see more and more requests on this type of product. So as a conclusion, I really would like to end on the image Martel explained at the very beginning. So that's like a chess game. That's really a global strategy. So hopefully you have now a better understanding of the concept of sterility. You know that you should choose terminal sterilization when it's possible, and do not overestimate your product sterility test. So thank you very much for your attention, and of course we are now available for the questions you could have. So we have a few minutes left, so we will do our best to answer them. And if we have no time for now, we will surely address them after the call. So thank you very much.
All right, great. Thank you so much, Anique and Martel. That was a really great, informative, in-depth talk. We do have time, like Anique said, for some questions, so let's start going through those so we can get to as many as possible. First up is more of a statement, really, than a question, but perhaps one of you could flesh it out a bit, give some more detail. And this audience member says, the assumption is that the 1 × 10⁻⁶ would be against a most resistant organism. So typical less resistant bioburden would be killed.
Yeah, that's, and do you want to address that, Anique?
No, go ahead, Martin, no problem.
Okay. Well, Anique referred to this a little bit on one of her slides, the slide with the pyramid, and where the idea is that statement is exactly correct. In most cases, the sterilization process is established with some conservativeness in mind. Many times that conservativeness is either higher counts than what you actually have, or where the microorganisms are of greater resistance than you actually have, or both. So that's a very good point, that in most cases the actual bioburden on the product is much less resistant. So the SAL of the bioburden in the product, it could be 10⁻20 or even smaller than that in many situations. That's a very good point.
Okay, great. Thanks, Martel. Moving along here, this audience member asks, can 0.2 micron filtration be considered as terminal sterilization?
Yes, so I will answer that one, if you don't mind, Martel. So microfiltration is not terminal sterilization, so that's a microbial reduction step. So definitely it helps, and it's usually used after aseptic assembly. But again, you cannot extrapolate or calculate any SAL for a filtration. So that's not a terminal sterilization. That's just an additional step to give more confidence in your aseptic assembly process.
Okay, great. Thank you.
And maybe I'll just add to that. As things are filtered, they're going into containers, and those containers, after filling with the filtered solution, those containers have to be closed. That is all happening in an imperfect environment, so there is that opportunity in many cases for some contamination event to occur, which is another reason why that's not considered terminal sterilization.
Okay, thanks, Martel. All right, let's see here. Next question. I think this is regarding the polling question the first polling question, where this person says the question was not written correctly. The first answer implies both methods are used. It should have said aseptic assembly or terminal sterilization. Do either of you want to address that?
Yeah, I can share a thought on that. Questions are always hard to write in a way that they cannot be misunderstood. So the intent behind that question was to make it clear that if there is not terminal sterilization in the process, then you cannot establish an SAL. If you were to do aseptic assembly and then afterwards you were to apply terminal sterilization, then yeah, certainly that would result in an SAL value. So thank you for that clarification. I want to make sure that distinction is very clear.
Okay, great. All right, moving along here. What is the difference between internal PCD and external PCD?
Yes, so I will answer that one. So the internal PCDs are really the PCDs that are placed inside the load, and typically this consists of a seed product. So we contaminate artificially a product and we place it inside the load. This one is really needed to assess the sterility assurance level, because that's the one we need to build our killing curve, if you remember the pyramid I explained before. The external PCDs are placed outside the load and are used to monitor routine processing, because it's much more easier to have the challenges placed outside the load instead of having to open the cartons and take out the internal PCDs. So during validation, we will demonstrate or show that the external PCDs are of higher resistance compared to the internal ones. And that allows us a more practical way to monitor our routine processing.
Okay, great. Thank you, Anik, for that explanation. Okay, let's see. We have a powder API where data indicates a gamma radiation dose of greater than 5 to 6 kGy creates impurities. Can a cycle with this low dose be validated for commercial production?
Yeah.
Go ahead.
Okay. So usually the answer is a yes with qualifications. So using standard sterilization methods, on one of Anik's slides, she showed that for radiation, you can establish a radiation dose, a sterilization dose, to about 8 kilogray minimum. That would be an SAL 10⁻⁶. So that involves very clean raw materials and processes. Now, if this API is in the finished product, and in the finished product state you want to apply 5 to 6 kilogray, you could consider that terminal sterilization, but you would likely have to incorporate the idea of an alternate SAL. You rationalize something closer to 10⁻⁴ or 10⁻⁵. If you want to sterilize it prior to the assembly of the finished drug, then yes, something like that, 5 or 6 kilogray to clean it up, could also be a valid option. In that case, it does not have to be 10⁻⁶, and those alternative values could be rationalized.
Okay, great. Thanks, Martel. Anik, did you have anything to add? I can move on.
No, I would just recommend the person who asked the question, if they want to know more, they can surely contact us for some trials. So we have really experts in that area, and the best way to answer the question would be to make some preliminary tests and to see what is the lowest dose that could be suitable for that specific API.
Alrighty, thanks.
And I'm going to add, Anik, with the other things that you mentioned to increase probability of sterilization, like with the inert environment or or refrigerated temperatures, which can improve product conditions after the sterilization process.
Yeah, definitely.
Okay, thanks, guys. Let's see. Next question asks, I understand it's relative and depends on the total process, but how cost effective is terminal sterilization over aseptic processing?
Yeah, this is a good question. I've had this discussion with friends in the industry, and you'll notice we're running a few minutes over, but this is something we love to talk about, so we're okay to go a little longer and try to answer these questions. Aseptic processing tends to be significantly more expensive, and it's because of the huge increase in controls and equipment and monitoring that has to occur, and even the validation part of that, it does significantly increase the cost. Estimates I've heard have been somewhere between at least 5 and 10 times more expensive than setting up for terminal sterilization, and perhaps even more than that as far as the difference goes. Because terminal sterilization does not mean you can be sloppy. You still need to be careful, but with terminal sterilization, you're expecting some microorganisms to be present, and so you do have that flexibility.
Okay, great. Thanks, Martel. Let's see, moving on. Could you describe the inert environment method that was mentioned?
Yeah, for that one, I'm not sure I really understand the question. Maybe Martel, you know more than me on that one, because
I think what they're asking is when we're talking about the mitigations that can be done to improve the likelihood of being able to sterilize. A great example of that is with radiation sterilization. If you have, for example, if it's a dry product and there is air in the packaging, excuse me, if you, prior to sealing that, whatever that package is, prior to sealing that, if you purge the air and have nitrogen or some other gas in there instead, a more inert gas, then that gives fewer things for the radiation to interact with, and can result in a more stable product with an inert gas rather than oxygen being present.
Yeah,
okay.
Thank you, Martel. I didn't realize it was for the radiation technology, so indeed you answered it very well.
Okay, thanks. Let's see, we have 2 more questions up here. One asks, what is PCD?
That's an easy one. So that's PCD for Process Challenge Device. So that's really the challenges we are using to make the validation.
Okay, thanks for the explanation. And we've got one more question here. What if bioburden level of product in bulk is up to limit? Should we be worried about sterility testing? Should I test more samples from the batch?
Yeah, an interesting question. So maybe we can answer both. I don't know, Martel, what you think, but definitely initial bioburden level is really important in terminal sterilization, especially because you want to show that your initial bioburden level is under control. So you need to make sure that you do not go over a specific limit. What is that limit? I cannot answer that. So that's really up to the manufacturer to define that limit and to make it to keep it under control. So based on that, you can validate your sterilization process, and for sure you can never bring to sterilization a product that is over that initial limit. So that's really the principle of having a reduction method. So keep in mind that sterilization is only a way to reduce the population of your microbes. So of course, the higher level you start, the higher probability of having unsterile product is there. So that's probably the first part of the question. Martel, maybe I let you answer the second part.
Yeah. So that on the other end of that, this is one question that we get quite often as a laboratory, where someone will call us and say, we had a raw material that came in with a really high bioburden. We want to perform some sterility testing to show that even though one of the components was high of our drug product, it still is sterile. And the problem with that is, once again, if the intent behind that sterility test is to prove that you still have 10⁻⁶, if it's a terminally sterilized product, then again, that's not going to give you any usable data. And the appropriate way to address that for terminal sterilization is to understand, okay, so we have this high value, we set the limit. First of all, I always ask, has the limit been established correctly? Many times, those limits, when I have companies look back and find where those limits came from, they say, well, we don't know where it came from. It's always been in place. But if it's properly documented, if you understand where the limit came from and why the limit is there, oftentimes if you understand something behind the resistance of the microorganism to the sterilization process and other factors, depending on the situation, then you can help understand what impact that might have on the product. If it's an aseptically processed product, then again, and in that case where there is no terminal sterilization in place, then a sterility test can help. But going back to one of the slides that Anik shared regarding the ability to find the contaminated unit out of the number you test is very difficult. So sterility testing would often be a component for aseptic assembly if you have this scenario, but there will be other things to consider as well that would be very dependent on the situation.
All right, great, thanks. Yep, so looks like we've gone through all of our questions here, so we're about 10 minutes after the top of the hour, so we're going to go ahead and conclude the presentation. If we didn't get to your question, Martel and Anik will have access to that, and they will follow up with you directly. But thank you again, Anik and Martel. It was a really great talk, lots of audience participation. That's always a great sign. And thank you very much to our session sponsor, Nelson Labs and Sterigenics. If you'd like more information on them, their info is up on the screen now here, so please visit them. And thank you, everybody in the audience, for your attendance and participation. As I mentioned earlier, at the top of the hour, this session will be archived for 1 year on our website, contractpharma.com. Thanks very much for joining us, and have a great rest of your day, everybody.